thank you very much, Trinity, for agreeing to give a Build a Soul seminar, and you can get started.
Right, I'm just gonna hide everyone here. Okay, I'm gonna assume that everyone can hear me and see my slides.
Yep.
Awesome. Already off to a great start. I'm Trinity Hamilton. I'm an associate professor in plants and microbial biology at the University of Minnesota. I did a postdoc at Penn State with Jen McElady and a PhD at Montana State with John Peters. Yeah, when people ask me what my lab does, I usually say something like we study weird phototrophs in weird environments as a very brief summary of what we do. And so Kate asked me to give this talk a while ago, and I thought immediately, of course, that sounds fun. And then more recently, Kate gave a really lovely talk in the BTI seminar series here at the University of Minnesota. And I panicked because I thought Kate is building cells, and that is clearly not what my lab does. But I took a look at some of the titles, and it looks like y'all explore quite a few things in the seminar series. I think I found a way to relate a couple of the projects in the lab to thinking about building cells or building functions and thinking about how evolution works in communities. Or specifically here, I guess we're thinking about photosynthesis, so more on the functional level. So I want to tell you a little bit today about how we use extant cyanobacteria to explore the evolution of oxygenic photosynthesis. And specifically, I want to tell you about two projects where we are characterizing sort of extant or modern cyanobacteria. One is thinking about cyanobacteria that live in early Earth analogs. So these are systems that are anoxic, so they don't have a lot of oxygen or any oxygen. And they're typically sulfidic or ferruginous, so ferruginous being rich in iron too. And we think these systems and the cyanobacteria in those systems can be good models for the evolution of oxygenic photosynthesis. And then the second project I'll tell you a little bit about is thinking about cyanobacteria that live at their environmental limits. And so in these types of studies, we're thinking about what are the dispensable functions of cyanobacteria. And then what we'd like to do is sort of go beyond that and think about where in a photosynthetic organism, both in terms of photosynthesis, chlorophyll biosynthesis, all of the functions that are sort of related to harvesting light and fueling cellular processes, where might evolution act specifically in all of those sort of genes and proteins that are involved in that process. So just to give you sort of a little background about why we're specifically interested in the evolution of oxygenic photosynthesis, that is because early Earth was anoxic and reducing. So what I'm showing you here is the percent atmospheric levels of oxygen over Earth's history. So we're going from 4.5 billion years to present on this graph. And this gray line shows you the increase in oxygen over time. So a couple of things to notice. This is on a log scale, and I'm not showing them here, but there are massive error bars on this plot. But what I want to draw your attention to is this first rise in oxygen, which we call the Great Oxidation Event. And that occurred at the boundary of the Archean and Proterozoic eons in Earth's history and was facilitated by the evolution of oxygenic photosynthesis, which first evolved in cyanobacteria sometime in the Archean. And sort of dating the rise of oxygenic photosynthesis is not something we do, but a lot of groups are thinking about specifically when oxygenic photosynthesis emerged. What we're thinking about in my lab is what oxygenic photosynthesis looked like when it first evolved. Presumably, it maybe wasn't very good at what it was doing and was potentially took a long time. Here we're showing, I don't know, maybe close to 1 billion years to sort of get really efficient at oxygenic photosynthesis, which today is this very generates a lot of energy. It's a very energetically favorable process. And so what we're looking for is environments that mimic these conditions. And so oceans at this time where we think oxygenic photosynthesis arose were probably largely ferruginous, so rich in iron too, with the potential for upwelling events in near-shore environments, leading to sulfidic conditions. So oxygenic photosynthesis evolved under conditions that are very, very different from Earth today. So we, again, characterize extant cyanobacteria that live in these early Earth analogs. We're looking for systems that are low in oxygen and have sulfide or iron too. So one of our favorite locations is Little Salt Spring. This is a karst cover collapse sinkhole in Florida. This is an aerial photograph of the sinkhole. It's about 78 meters wide and 78 meters deep. It formed before the end of the last ice age. And so this is what the sort of a schematic of this hourglass shape of the sinkhole. So we have dissolved oxygen is present in the upper oxic zone of the sinkhole and then goes to below detection limit around 10 meters. And we also have sulfide present throughout the water column of the sinkhole. So there's this debris cone, a lot of organic matter, an organic matter rich system. So we have sulfate reducers producing sulfide, resulting in sulfide being present throughout the water column. So there's eukaryotic algae that live up here near the surface, these chara. They are producing oxygen, doing sort of normal oxygenic phototroph things. What we're particularly interested in this system are these pinnacle mats that live sort of in the lower basin, and then down along the sort of neck of the sinkhole, in this part of the sinkhole where there's sulfide and little to no oxygen. And this is a couple of pictures of what those pinnacle mats look like to the divers. So that was also the picture I should have said on my cover slide, the first slide of the talk. And so it's really sort of this very thin, but really cohesive purple reddish mat that blankets this upper basin and then down into the neck. So it's a significant amount of sort of thin mat, if you will, that is dominated by cyanobacteria. So stepping back for a minute and thinking about what an oxygenic phototroph looks like in terms of machinery. This is sort of a typical oxygenic phototroph, which has photosystem 2 and photosystem 1. So it has two photosystems or reaction centers that work in concert, in parallel, to photooxidize water and fuel cellular processes. So the photooxidation of water happens in photosystem 2 at the oxygen evolving complex, which is this little inset that I'm showing here. So again, oxygenic phototrophs have two photosystems. They work in parallel. Anoxygenic phototrophs have just one reaction center. Reaction centers and photosystems are the same thing. Reaction centers are what we call these systems in anoxygenic phototrophs, and we call them photosystems in oxygenic phototrophs. Anyway, anoxygenic phototrophs have reaction center 2 or reaction center 1, so they have just one reaction center. They're still harvesting light to fuel cellular processes, but they're not evolving oxygen. Most of these oxidize sulfide, iron 2, or hydrogen. And some of them are autotrophs, some of them are heterotrophs, some are facultative autotrophs. So lots of metabolic diversity within the anoxygenic phototrophs, as opposed to cyanobacteria that live in a multitude of different environments, but in general, they're performing this photoautotrophy using water. So what we want to think about in the lab is how oxygenic photosynthesis was first built and what those organisms look like in terms of their ecology and photosystem content. So again, these are our sort of extant cyanobacteria with these two photosystems. But to get to these two reaction centers working in parallel, we had a series of gene duplication events that led to the two reaction centers. We have to get two reaction centers in one cell. So again, anoxygenic phototrophs have just one reaction center in their cell. And we also have to give these reaction centers or photosystems working in concert. So this is a highly paralleled and controlled process that's happening together. And there's a bunch of other things we can think about and other labs are thinking about in terms of the gene duplication events. So this can be really useful for molecular clocks. There's also a whole bunch of accessory proteins that have been added specifically to photosystem 2 to increase activity of photosystem 2 and stability of the oxygen evolving complex and just make this process more efficient. And so what we're specifically thinking about, though, is if a one photosystem cyanobacteria is a plausible model for the evolution of oxygenic photosynthesis. So we have anoxygenic phototrophs that only have one reaction center, And then we have these cyanobacteria with 2 reaction centers. So is there an evolutionary step where you got a cyanobacteria that was using 1 photosystem? And this has been proposed before. This is a little schematic proposed by Allen and Martin in 2006, where they said, okay, this is your extant cyanobacteria. What if an evolutionary step of getting these 2 photosystems working in parallel is to have them differentially expressed? So they're in the same cell, but they're not yet working in parallel. You have reaction center 2 that's harvesting light energy, but it's just making ATP. And then you have photosystem 1, which is oxidizing sulfide and using light energy to fuel cellular processes. So there's a couple of examples of cyanobacteria that do this process, this sulfide-dependent process. And that's sort of what led Allen and Martin to this model, is that we already have examples of cyanobacteria that do this, but what if they could differentially express their reaction centers in their evolutionary history? So coming back to our little salt spring, we did a bunch of work in this De Beer paper where we characterized anoxygenic and oxygenic photosynthesis in situ over several 24-hour periods. And what we learned from those studies is that oxygenic photosynthesis only occurs for about 4 hours a day. During the rest of the photoperiod, so the rest of the time the sun is up, if you will, photosynthesis is anoxygenic in the sinkhole. And so that led us to sort of ask whether or not this strain Hansonii, which we isolated from the sinkhole, could operate under sulfidic conditions. Could it do oxygenic photosynthesis in the presence of sulfide, or could it do facultative anoxygenic photosynthesis? And this is just the pinnacle mats I sort of showed you in the sinkhole. This is how this organism grows in situ, or sorry, in the lab. It still likes to make these weird little small pinnacles. It likes to grow on sand. But I also like to show this figure because this organism clearly does oxygenic photosynthesis in the lab. So if there's no sulfide around, it will happily do its 2 photosystem photooxidizing water and producing oxygen as its byproduct. So we isolated Hansonii and we characterized the strain in the presence of sulfide. And this was a large and collaborative project with some folks at the Max Planck Institute, namely Judith Klatt and Dirk De Beer. But we added sulfide and asked what happens to oxygenic and anoxygenic photosynthesis. So here I'm showing gross photosynthesis over time, and the closed triangles here are oxygenic photosynthesis, and the open squares are anoxygenic photosynthesis. So as soon as we add sulfide, we inhibit oxygenic photosynthesis, and we see anoxygenic photosynthesis proceeds until all of the sulfide is consumed. So this organism Hansonii does anoxygenic photosynthesis, but also sulfide inhibits oxygenic photosynthesis. And this is very common. We see this in plants and algae and other cyanobacteria that sulfide binds to photosystem 2 and inhibits oxygenic photosynthesis. We did a bunch of experiments. I'm just going to show 2 here, the previous one and then this one, where we're looking at gross anoxygenic photosynthesis compared to oxygenic. And I'm representing that as a % of that activity. And we do these measurements over a bunch of different sulfide concentrations and at high light and low light. And so in the low light condition, we see anoxygenic photosynthesis around 60% of oxygenic photosynthetic activity. If we turn up the lights a little bit in these orange squares, we see that, in fact, anoxygenic photosynthesis can be about twice as high. The activity can be twice as high as oxygenic photosynthesis. So these organisms not only perform anoxygenic photosynthesis, it depends on light and sulfide concentration, and the rates can also exceed oxygenic photosynthesis. So this seems like an activity or a physiology that could be successful in a natural environment. So this tells us or indicates to us that an early evolving 1 photosystem cyanobacteria is plausible. Hansonii performs anoxygenic photosynthesis. Again, this is a photosystem 1 dependent process. It responds to environmental signals, and the activity that we see is consistent with conditions in situ. So this doesn't seem to be an artifact of something we're doing in the lab to sort of favor this activity. And then rates can be comparable to oxygenic photosynthesis. So this seems like a way that an organism could support itself in situ. So we haven't actually shown differential expression. So if we come back to this model put forward by Allen and Martin of the proto-cyanobacterium, we know this is a photosystem 1 dependent process. That hydrogen sulfide is oxidized to elemental sulfur, and these electrons are dumped into the cycle just above photosystem 1, and that this organism fixes CO2. So it does facultative and oxygenic photosynthesis. What we haven't yet shown is what is happening with reaction center 2 or photosystem 2 in our strain Hansonii. And so that's what we're working on now. We do see decreased amounts of photosystem 2, but we're working on whether or not that is actually differential expression, or if the organism is still making photosystem 2 and it's just being inhibited by sulfide. And so, as I mentioned earlier, sulfide will bind photosystem 2 and inhibit oxygenic photosynthesis. And so that's fine and interesting, but for thinking about sort of the evolution of this process and whether or not you could have had an organism with just one photosystem or just one photosystem operating at a time, it's important that these photosystems be differentially regulated or regulated by different environmental signals. And so that's what we're working on next. I should mention, well, 2 things. We have isolated a second cyanobacterium from the same sinkhole, and it also does this activity. So we're really excited to compare these 2 organisms. We have just sequenced the genome of the second one, so there's some really exciting things we can do in terms of comparing them. But I'm also, as a shameless plug, looking for either a postdoc or a lab tech to sort of continue on this project. So if you know of anyone who's excited about photosynthesis and tinkering around with microsensors, please send them my way. Okay, the second thing I want to tell you about is thinking about extant cyanobacteria at their environmental limits. So again, we want to identify things that are dispensable to organisms. So if they're sort of stressed, and I should say we're not looking for subtlety in this in terms of environmental limits, we're really sort of at the edge of where these organisms are surviving, and I'll show you that in a minute. But so we want to know, as that environment is potentially stressful or causing different photosynthetic machinery or other sort of cellular responses to be more upregulated in terms of sort of repair functions and things like that. Are there other functions that these organisms will just get rid of to allow them to sort of maintain other parts of their system? And then again, we want to understand specifically where in photosynthesis evolution acts. So I said we weren't looking for subtlety. We are in this particular, the study that I'll show you today, we're thinking about the upper temperature limit of photosynthesis, which occurs at 72 to 73 degrees Celsius. So photosynthesis just does not occur above this temperature in any environment that we've observed on Earth. In our lab, we call this limit the fringe. And so this is a hot spring in Yellowstone National Park. This in the center here is the source water. So this is probably somewhere around 92 degrees Celsius, boiling in the park. And then this is the outflow channel of that hot spring. And so as that hot water flows down the outflow channel, it cools simply due to physical processes of your source being here and this water being farther away. And what you can see is this sort of white silica-rich armored channel starts to become greenish, orangish here as you get farther and farther down the outflow channel. And so that would be your indication of where that sort of 72 to 73 degrees Celsius cutoff is as that water is cooling. And so this greenish, orangish stuff is photosynthesis or photosynthetic organisms due to the pigments they make. That's where that color comes from. So anyway, we call this the fringe. And photosynthesis does not occur above this temperature. And so we're particularly interested in what this upper temperature limit does to phototrophs in that system. This is just one more example. It's hard for me not to show a million hot spring photos, but I'll just show you 2. This is just another example where your source spring is going to be over here somewhere and the water is flowing in this direction. And so you have this hot water out here in sort of the main pool. And as you get to sort of the shore, if you will, or the banks of the stream, that water cools enough to allow phototrophs to occupy that niche space. So here you're seeing that sort of orangish, reddish, greenish mat of phototrophs. The reason that I like to show this or this a figure like this or a picture like this is because there's life out here in the outflow channel. So these filaments out here along these little silica-coated rocks. This is life living out well above 72 or 73 degrees Celsius. So microbial life can definitely live above this temperature limit. It's just photosynthesis that is limited by this 72 to 73 degrees cutoff. So if you were to take a walk down a hot spring outflow channel, so if you're standing at the source over here at high temperature and you walked downstream, down the outflow channel, here at this zone, or if we go back to here, if you're up here in this sort of hottest region of this hot spring where you're first starting to see phototrophic mats, you would encounter the fringe, and this fringe would be comprised largely of organisms, specifically cyanobacteria within the Synechococcus AB clade. This might not be your jam, taxonomy of cyanobacteria, but the idea being that most of the cyanobacteria that you see within this, certainly as you approach that upper temperature limit, are sort of limited to one genus or lineage. So these are our main examples of high temperature cyanobacteria are within this lineage. And Dave Ward and Don Bryant, Dave Ward at Montana State University and Dave Bryant at Penn State have done decades and decades worth of work in Yellowstone National Park, specifically in mushroom and octopus springs, to characterize these organisms at the fringe and downstream. Octopus spring might be familiar to you because that is where Taq polymerase was first discovered in an organism isolated from octopus. Anyway, so Dave and Don have described these what are called ecotypes of cyanobacteria across this temperature range. So these are closely related yet ecologically distinct organisms. They occupy distinct temperature ranges and vary in their optimal growth temperature. So they are living in separate niche space and having these distinct temperature ranges, but at the typical cutoffs for what we would call species, these organisms are typically too closely related to be delineated into species. And so instead, Dave and Don and others have proposed that these be called ecotypes because they can't be delineated specifically into species, but they are occupying distinct ranges. Okay, so what we were specifically interested in is, if you walk down this fringe, we have A prime, A double prime, A prime, A and B prime, and you can sort of walk farther and farther down the alphabet as you get farther and farther downstream with the ecotypes. But what I wanted to draw your attention to is that as you get in sort of up in increasing temperature, the temperature range of these high temperature ecotypes decreases. And so we wanted to know what is leading to that decrease in temperature range of these high temperature ecotypes. And so what we specifically asked is what functions might differentiate A primes. We're specifically looking at A prime in this study, which is sort of the second highest temperature cyanobacteria, if we don't consider it A double prime. So what we did is we sequenced a bunch of metagenomes from different locations in Yellowstone National Park. And then we recovered what we call MAGs, these metagenome assembled genomes. So we don't have these organisms in culture. We're using a purely sequence-based approach to analyze their genomic content. And so what we did is we sequenced a bunch of metagenomes, recovered the metagenome assembled genomes, and then I'm going to show you those MAGs, as I'll call them, across temperature and pH space. And so in the sort of greens and pinks are going to be anoxygenic phototrophs. And then in the warmer colors are going to be our cyanobacteria. And specifically what we're interested in is the Synechococcus A prime like ecotype. And when we looked at the relative abundance of the MAGs across this in our different sites across this temperature and pH space, we observed exactly what we were supposed to see. So in this tealish color are our A prime MAGs, and the larger the bubble, the higher percent relative abundance. So A prime MAGs are more abundant in our sort of sites that are near or at the upper temperature limit. And then the only MAG that we see below this sort of upper temperature limit is around 64 degrees Celsius. So we have our A prime. There's a really interesting story, I think, to think about in terms of anoxygenic phototrophs here at the fringe, but that is a story for another time. So we see A prime is prevalent in our highest temperature sites. Again, exactly what we were supposed to see. But what we're interested in is their genomic content. And so what I'm going to show you is a figure where we analyze the genomic content of our different MAGs with some reference genomes The main thing that we observed is that ecotypes differ in a small fraction of their genomic content. So a lot of their genomic content is shared, but then there is sort of small amounts of the genomic content that is not. So this is a sort of a genome circle of a bunch of our mags and a handful of reference sequences. So each circle here is a genome and represents all of the content of that genome. And where the bars are darker is where a gene or a gene family is present. And where the bars are lighter is where a gene family was not detected in that genome. And then we're looking at B prime, A, and A prime mags here, but I'm just going to talk about the A prime like mags. And so we have this core and auxiliary core genome where we see a lot of these sort of darker bars where these genomes are sharing a lot of content. And then we get out here sort of farther away in the genomic content where we start to see some differences. So we see places where A has some extra genes, B prime has some extra genes, A prime has a handful of extra genes compared to these other two. But what we're specifically interested in is this area, this large area where A prime is depleted in genomic content. So then we asked specifically, what are some of the functions that differentiate these high temperature?
I don't have an answer for that. Is there something else I can help with?
Siri is asking me what I want to do. Hopefully you didn't hear that. Okay.
I'm not sure I understand.
I don't know what's going on with my Apple Watch. Okay, so we're specifically interested in what functions differentiate the highest temperature cyanobacteria. And so A prime lacked about 250 genes compared to these lower temperature ecotypes. And some of these were what we would consider dispensable oxygen sensitive enzymes. So specifically, A prime lacks nitrogenase and this dark operative protochlorophyllide oxidoreductase. It's a very fancy way to say this enzyme that makes chlorophylls in the dark. And so both of these enzymes, nitrogenase and DPOR, are oxygen sensitive. Nitrogenase is the enzyme that allows some bacteria and archaea to fix dinitrogen out of the atmosphere into ammonia. It's a very complex and energy intensive process and includes a lot of oxygen sensitive enzymes. And so what I'm showing you here is a heat map of gene occurrence. So we've just binned all of our genomes now for B prime, A, and A prime. And then sort of the darker the color here, the higher abundance those genes were in those mags. And the lighter the color, those lower abundance. And so if we look across our A prime, we see that there was no light independent P-chlide reductase. This is this DPOR. Again, this is the ability to make chlorophylls in the dark. And there's no nitrogenase or nitrogenase associated proteins. So these NIF proteins. There's also a couple of other things missing, like urease and associated proteins. But we were specifically interested in the fact that there was no nitrogenase detected in our genomes and no ability to make chlorophyll in the dark. And we simply wanted to ask, does that make sense? So we don't, as I mentioned, we don't have these organisms in culture, and this is purely a DNA-based environmental observation that we're making with this genomic content. But we have a lot of information about the hot springs. We know a lot about the geochemistry, pH, temperature. And so we can simply ask, does it make, environmentally, would it make sense to not have these or to get rid of these enzymes? So A prime encodes an ammonium transporter and a full suite of nitrate assimilation genes. So it doesn't seem to be lacking for ways in taking up nitrogen, fixed nitrogen from the environment. We don't see a lot of ammonia in these systems, and that's typical for ammonia D gases around this temperature and pH. So these systems can be limited in fixed nitrogen, but we do see in most of the sites where we found A prime, we have micromolar amounts of nitrate. So we assume this comes from sort of recent precipitation events in the groundwater or leaching from nearby environments. So there is some amount of fixed nitrogen. A prime also encodes this protochlorophyllide oxidoreductase that is not oxygen sensitive and operates in the light. So it can clearly still make chlorophylls and it can assimilate nitrogen from its environment. So we, again, are thinking about why, I mean, this is potentially a nitrogen limited environment at some point in time, but oxygen sensitivity shouldn't be a concern for these organisms. Nitrogenase and DPOR are specifically oxygen sensitive enzymes. We only found A prime in filaments. And so there's a couple of pictures of what these filaments might look like in situ. And these systems are high temperature, which means they have low oxygen solubility. And so this is my sort of view of what a filament might look like if I had a video. These things are sort of waving around out in the channel, out in the outflow channel of these hot springs, and oxygen is not accumulating the way it would in mats. And so if we're thinking about how our systems compare to sort of Dave and Dawn's mats in mushroom and octopus spring, in these mats or in mats anywhere, you can have sort of high amounts of oxygen accumulating during the sunlit hours. But in these filaments, that really shouldn't be the case. And so we think that the loss of DPOR and nitrogenase is simply because they are dispensable, that these organisms sacrifice the ability to make chlorophylls in the dark and rely solely on environmental nitrogen, fixed nitrogen. And so we haven't yet sort of linked this to high temperature stress, but it is interesting that they're ditching things that are purely dispensable. And that it is probably due to their dispensability rather than their oxygen sensitivity, at least based on what we know about the environments that they are found in. Okay, so hopefully today I've convinced you that a one reaction center archaean cyanobacteria is plausible as a model, as a step in the evolution of oxygenic photosynthesis. So thinking about cyanobacteria that either had just one photosystem or was able to sort of use the photosystems for different processes at different times and or regulate them differently based on environmental cues. And then we see that high temperature leads to genome streamlining in cyanobacteria. And we're super excited about thinking about, is this specifically a high temperature phenomenon where you sort of drop some of these dispensable enzymes that are sort of large, oxygen sensitive, energy intensive processes and potentially hard to maintain because specifically due to high temperature stress. And then we're following up on these studies to think more specifically about where evolution is occurring at the gene or protein scale. And these are the folks that actually do the work. I'll highlight specifically Amanda, Caleb, Katie, and John, who are undergrads that contributed to these projects, and the other folks that have led and or contributed here. We got some money from NASA and NSF to do this type of work. And then specifically the cyanobacteria in Little Salt Spring has been a large collaborative project with folks from Penn State and the Max Planck Institute, as well as a bunch of science divers that have helped out with that project. So that's what I have for you today, and I'm happy to answer any questions if there are any.
Thank you so much. That was amazing. There is a lot of questions in chat. Can you see chat?
Yeah.
If you could read a question out loud before you answer it, that's going to go for the recording.
Yeah, perfect. Okay, let me scroll up here. Would it be possible to engineer a photosystem that works above the fringe? I don't see why not. I have many thoughts about this, but it seems like the upper temperature limit is probably due to potentially due to radicals in the carbon fixation pathway, and rather than specifically due to the photosystem. So I think what we've been playing around with a little bit is thinking about if you could just put a cyanobacteria in the dark at higher temperature, what happens, and or if you could grow a photoheterotroph above that temperature limit. So it's interesting that that no photosynthesis happens above the higher temperature limit because anoxygenic phototrophs are living with cyanobacteria at all of those mats that I showed you. And so they only have one reaction center. And some of them use the RTCA and or are photoheterotrophs. And so we're interested in knowing why that upper temperature limit exists for them as well. But I like this idea of engineering a photosystem. It would be more thinking about, I think you would have to think about whole cell and whether or not it was specific to carbon fixation. But in general, I like the idea of engineering the photosystems. Because of the temperature limit of chlorophyll, the key is to make a high temperature pigment. No, oh yeah, good point, Lynn. Yes, it was first isolated from mushroom spring, which is about a 5-minute, yeah. Taq polymerase from mushroom spring. So Lynn brings up a good point there. Mushroom and octopus spring are very close to one another in Yellowstone. Do you think the photosystem always had this temperature limit, even back when Earth was warmer, or was there another type that got lost over time? I think it's probably always had this temperature limit, because why not just go there, go above this temperature limit in hot springs today? If it truly is a photosystem process that is inhibited and or carbon fixation, yeah, evolution has done a bunch of wonderful things, so I assume it could go above this temperature limit, but I don't have a great answer for that. Do you know if there is any effort in engineering these gene clusters for photosystems for use in synthetic contexts in liposomes or completely in vitro? Yes, for sure. I think I don't know about sort of in really minimal systems, but I'm fairly certain that photosystem, maybe it's the whole photosynthetic machinery, has been, they're trying to express it in E. coli, and they might have done it already. I'm not exactly sure. I should follow up on that research, but there is certainly a lot of interest in synthetic systems and Rubisco. There's a lot of work in sort of synthetic Rubisco and trying to make Rubisco more efficient as well. Does the temperature limit for photosynthesis depend in any way on other environmental conditions like salinity or pH? Does polyextremophile have to sacrifice some temperature tolerance for salt tolerance, for example? Salt tolerance potentially, but pH absolutely. And so when I showed you, I think you can still see my screen, this slide. The upper temperature limit, oh no, where'd it go? I lost it.
We see your desktop now.
There we go. Why does it keep doing that? I don't know. This figure, so we have the upper temperature limit of 72 degrees Celsius is actually only observed in alkaline pH. As you go to lower pH, the upper temperature limit drops. So the upper temperature limit is linked to pH somehow. And in acidic systems, the upper temperature limit is around 56 degrees-ish Celsius. And those systems are dominated by algae. So we don't see a lot, if any, cyanobacteria at the lower pH. But that the upper temperature limit is lower at acidic pH, and I don't think we fully understand exactly why that is yet. What about low temperature? What's the low temperature limit for photosynthesis? Great question. So the lab wears many hats, and then the other part of the lab is studying snow algae, so algae that live on snow and ice seasonally. Those algae are operating at 0 degrees Celsius. I'm sure there are examples of photosynthesis below 0 degrees Celsius in slightly brackish water. I'm thinking of some of those ice, either under ice algae that have been studied. And so there is a lower temperature limit. I'm not exactly sure what it is off the top of my head, but as someone mentioned earlier, it has to do with salinity. So there's probably a point where this salinity is impacting photosynthesis in a way that doesn't allow it to occur. So typically we say if there's liquid water available, there should be photosynthesis, but that's not entirely true because it is also linked to salinity. I should say that on our snow samples, we don't see cyanobacteria. So they live in alpine streams at, just above freezing, but not a lot on snow surfaces for whatever reason. Did anyone reconstitute the whole photosystem in vitro? Yeah, so someone else is asking this as well earlier, and I think you're right that it's still difficult. I'd have to go back and look at some of those. I don't know if I've seen it in a talk or if it's actually published yet where people are trying to do this, but yes, I think you have to do multiple steps to build the membrane and build the systems. I don't think you can yet do it in vitro in one sort of step yet. But yes, the membrane part is definitely tricky. Could it be possible to replace chlorophyll with another more temperature stable complex? Nature never did, but we could imagine engineering it from the chemical point of view. Yeah. Replace chlorophyll. I don't see why not. It's not my sort of expertise to think about engineering in that way. But from what I understand in terms of synthetic biology on photosynthesis, most of it is thinking about sort of building the whole photosystem in vitro and making Rubisco more efficient. And so I don't know if that means that people have tried synthetic chlorophylls or synthetic chlorophyll-like pigments and they haven't worked, or if it's also included in those studies and I just haven't sort of looked at them close enough. But yeah, I think why not? Maybe it has to do with, I guess they're probably, I think in terms of high temperature photosynthesis, I don't think there's a large or a significant bioengineering. I don't think we need photosynthesis to work at high temperature in terms of sort of biosynthetic processes or thinking of engineering, bioengineering. And so I think it's more sort of from basic research and thinking about how, how photosynthesis, what limits photosynthesis. But I think most of those synthetic biology studies are focused on improving photosynthesis or making it more efficient at sort of, I guess I would call it more regular temperatures, if that makes sense.
Yeah, I actually have a question. I see you went through all the chat questions already. So I'll take the opportunity to ask a question. When you mentioned improving photosynthesis, I'm not sure the numbers, but I kind of remember reading that photosynthesis is actually, Rubisco is actually a very inefficient enzyme. Is that what most of the efforts are at, trying to increase that efficiency?
Yes, from what I understand, that is a large part of thinking about improving photosynthesis, and probably a lot more. I think those studies are being done in cyanobacteria, but I don't know if it's potentially for biofuels, but also probably thinking about how to make that those tractable in sort of crop plants, if you will. But yes, there's a lot of interest in increasing Rubisco efficiency.
I'm surprised because I would think evolution would have done more on it, because I could imagine that would be an immediate fitness boost if you are more efficient at photosynthesis. So
yeah, I think the question of Rubisco being so abundant and so not great is an interesting philosophical question, I guess.
Kind of like the ribosome. Oh, Len, I see you. You have your hand right.
Yeah. Let me jump in on it. Someone who did PhD on Rubisco. Yeah, it's inefficient in that it's not efficient at using photons. Who cares if you waste photons? There are tons of them. And Rubisco is also used for other things like nitrogen storage, so it doesn't have to be overly efficient because it's got other purposes. And I've heard about people working on the increasing efficiency of Rubisco, thinking that that's going to solve agricultural problems for 40 years. And even the people who've been working on it have come to the conclusion, and generally the older people, that it's not worth it, but yet another generation is going to try. So I'm not sure that really, yes, it's inefficient. Who cares about photons? And that may not solve your problems anyway because of other uses. So that would be my summary.
Fair.
I see Vinod has a comment too about.
Yes, studying photochemical synthesis of abiogenic photoautotrophic protocell-like microstructures, which they sterilized aqueous mixture of some inorganic substances. That sounds promising and cool.
Hello, Trinity. So nice, very nice talk.
Thank you.
This is Vinod from Chicago. And this work was initiated by late Professor Krishna Bahadur in India. And he synthesized some protocell-like microstructures using some inorganic and organic substances utilizing sunlight as a source of energy. This mixture to sunlight, and he could observe the photochemical formation of protocell-like microstructures in the mixture. And when he examined these microstructures, they are spherical in shape, having a definite boundary, and they show multiplication, growth, and some metabolic activities. And abiogenesis, a lot of work has been done on abiogenesis of biochemicals also. What I mean to say that we have a system as a boundary wall for charge separation. And the work we are doing with at King's College with Professor D.O. Hall. And I introduced a photosynthesis-like material in the system. And we used different level of oxidized sulfur, and it is liganded with iron. And he used EPR, and he detected the presence of redoxin-like material in the mixture. And we could see that under certain specific conditions, these protocell-like microstructures can decompose. They show photolytic decomposition of water. And this work was taken up by a group at Ames. Dr. Adolph Smith, he was there. He passed acetylene over head the mixture. And this acetylene was reduced to ethylene and finally to ethane. Then one group, then Professor Claire Folsom at Hawaii University, he used heavy water, D2, and this acetylene becomes CHD, indicating that hydrogen is coming by the photolytic decomposition of water. And in subsequent experiments, we used C14 and N15. We used labeled nitrogen and carbon, and we could see that it is coming by the hydrogen is coming by the photolytic decomposition of water. So, but we further need to characterize the reaction center of this mixture, because our mixture, it contains so many ammonium salts, some transitional elements are there, certain biological catalysts are there, and the formaldehyde is the source of organic carbon. We have a system for charge separation. We have a photoredox process in the mixture. Under certain condition, it is a redox reaction. And some enzyme like you said that oxidoreductase, and I was also working in the presence of ATPase-like activity. So for photosynthesis, we need a mechanism for redox process, electron transfer process, and the charge separation process. So it seems to be an interesting system, but we further need to characterize the reaction center. But I very much appreciate the work you are pursuing, and I agree to it. Thank you very much.
Thank you. Yeah,
If
we're
possible,
very much.
I will be pleased to have any collaboration if we can. I'm in Chicago right now, and I can send you some. Please send me your mail. I will send you some references.
Sure. Yeah. Excellent. Thank you.
That will be
Yeah.
interesting.
Thank you. Oh, and Trinity Taysok has a question too in chat.
I was wondering what would be your perspective regarding super efficient photosynthetic bacteria that would solve the climate crisis to some extent, but there would be biocontaminant or biocontainment issues, super efficient photosynthetic bacteria.
Yeah, I could imagine if something super efficient at photosynthesis, biocontainment would really have to be strict, because that would be.
Yeah, to add something, so, the genetically modified bacteria potentially release or deploy in the real environment to solve the whatever climate crisis. That might be super cool, but at the same time, one concern I might have, those bacteria might dominate entire population, because that is the super efficient compared to native other photosynthetic bacteria or organism. And then I just wondering whether you have been thinking about this kind of potential scenario, or what would be your perspective because you are expert in this area?
I haven't been thinking about it in that yeah, I mean, I guess the closest thing that we've thought about in the lab is sort of earlier periods in Earth's history where we have sort of skewed CO2 to oxygen ratios, and what Earth might look like under there. I think there's always going to be a tipping point, right? So without biocontainment, the impact they're going to be potentially super efficient under the conditions now, but eventually those are going to tip as Earth has in past between levels of CO2. And so I think it's a tricky question that I haven't thought a ton about. I mean, people have thought about there's the plant more trees initiatives or whatever. And so I think those are less substantial on a global process or a global scale. But I guess I would wonder like how efficient your phototrope has to be in terms of volume for this biocontainment issue, for sure. But yeah, it's definitely a fun thought exercise.
Yeah, actually, we are making some. Actually, we already demonstrate kill switch to minimize any risk of genetically engineered microbes, and then we're actually transferring that into photosynthetic bacteria, including cyanobacteria.
Yes.
So if you are interested, just let me know. I mean, I'm happy to chat with you.
Okay, cool. Yeah, it would be fun.
Yeah, a great talk. Thank you.
Thanks.
Awesome. Thank you very much. I don't see any other questions, so I just want to say thank you so much, Trinity, again.
Yeah, super fun. Thanks
And-
for having me.
And thanks everyone else, and have a great week. Bye.